Okay, so we have all these phytocannabinoids like THC and CBD and CBN, that act at, what have now been identified as the CB1 and CB2, receptors in the brain and body. Right? These are endogenous of our own body. We make the receptors. And this is very much like the opioid story, right? Where you had, plant-derived chemicals, as you do in cannabis, that have, very well-known effects, the effects that have been known for millennia, in many cultures around the world. And then, when you look in the brain and the body, right, in the nervous system for, where these drugs are acting, well, you identify, specific receptors, and, the opioid receptors that we discussed, right? And for cannabis, we now know for these phytocannabinoids, they're acting at CB1, receptors, which are largely localized in the central nervous system, and CB2, which are more, commonly found in the peripheral nervous system and in areas of the immune system, like the thymus and the spleen. And so if we have receptors for cannabinoids, the phytocannabinoids like THC and CBD, well, why are they there, right? That's the question. Like, what neurotransmitters are actually acting at these endogenous, receptors? What endogenous cannabinoids do you produce? And two of them, there's been a number of that have been identified actually, but two very important ones. One has a very evocative name, poetic name. It's the Sanskrit for the joy without which the world would not exist. It's called anandamide, or AEA. And the other one is more prosaically referred to as 2AG. And what's fascinating is that these are lipids, and they cannot be stored in vesicles. They would leak out of a vesicle. So it turns out that the precursor for anandamide and 2AG is a chemical backbone called arachidonic acid, which is a constituent. It's a component of those phospholipid bilayers. And another really interesting aspect of these endocannabinoids is that they are actually released. They're synthesized, we're going to see, from arachidonic acid. There's got to be a synthesizing enzyme that'll convert them into 2AG or anandamide. But they're actually going to be released post-synaptically. So the post-synaptic cell, like the dendrite of the next cell, is going to actually release the endocannabinoid, and it will go back. They call this retrograde signaling, and it'll act at CB1 receptors in the brain, let's say, that'll be located on the presynaptic terminal. So this is going to be one of those heteroreceptors, right? And what happens is the depolarization of the postsynaptic membrane will result in the activation of a synthesizing enzyme in that postsynaptic cell, and that will cleave or cut out, the anandamide or the 2AG, from the arachidonic acid, which is present in the phospholipid bilayer of that postsynaptic cell. And then the 2AG, let's say, will go back and bind to CB1 receptors presynaptically. And what it tends to do is it tends to inhibit, the further release of neurotransmitter. These endogenous, cannabinoid neurotransmitters have been proposed as important, constituents in a sort of system for forgetting, for letting things go, actually. And, it's a very unique, not a classical neurotransmitter we're talking about it here at all, right? It is not stored in vesicles. It is not released typically presynaptically, but postsynaptically, cleaved out by, enzymes, upon the depolarization of that postsynaptic cell, the next cell in the, the network. It works its way back retroactively to act at receptors that are presynaptically localized, right? These CB1 receptors, and usually has an inhibitory effect on, further transmitter release. So very interesting, endogenous cannabinoid transmitter system, and a lot of, research is ongoing, elucidating additional endocannabinoids and additional sort of actions, that these chemicals may be taking.